Hello again, and welcome to the COVID-19 pandemic briefing brought to you by Pirate Biotechnology, Morro Bay High School. So, where we left off last time is with the hammer and the dance, which is what the healthcare experts are recommending that we do right now, which is use 3 to 7 weeks to hammer down the rate of infections. And then, meantime, after that, work out solutions in order to keep the infections at a manageable rate while we're working on a vaccine. So that's the plan. Hopefully, that's what we do. So that was part one. Part two, we're going to look at what even is coronavirus. So here we go. So SARS, coronavirus 2, is called coronavirus. They call it SARS because it's the second coronavirus that we've discovered that has jumped from bats to humans through an intermediate organism. And SARS stands for severe, which means hardcore, acute, which means all of the sudden, respiratory, which means you're breathing, and syndrome, which means something's wrong with it. So SARS means something that comes upon you all of a sudden and messes up with your breathing. So SARS coronavirus 2 is called coronavirus, as opposed to SARS coronavirus 1, which happened in 2002, mostly in Asia, but quite a bit in Canada also. So coronavirus, or SARS coronavirus 2, causes coronavirus disease 2019. And if you look at what I put in bold right here, COVID-19, coronavirus disease 2019 is called COVID-19 for short, which surely you've heard of. That's the disease caused by coronavirus. And here we are to talk about what coronavirus is. So here's what it is right here. We've got this particle right here, and the particle is made of proteins, and then the proteins are embedded in a phospholipid bilayer, which is the same thing that all of our cells are encased in. It's kind of like a fat. And then inside of that particle is RNA, which is very similar to DNA. How similar? Well, let's check it out. So DNA is double-stranded, and the cool thing about DNA is that it's super robust. You can take some DNA and smear it on the nose cone of a rocket and fire that rocket up into space and then bring that nose cone back and catch it, and then you can swab that nose cone and you'll get that DNA back again, still intact. It's super robust. RNA is not as robust as DNA, but it's easier for cells to work with because these nitrogenous bases here are already exposed. In DNA, when it's double-stranded, the nitrogenous bases are protected inside of this sugar phosphate backbone, and an enzyme needs to come and split the thing open to make it look like this before the cell can read the instructions on it. So DNA versus RNA. DNA is what our cells have for genetic instructions, and RNA is what coronavirus has for genetic instructions. This is a map of the genome for coronavirus. Each one of these colored bars represents one gene, and each gene codes for one protein. Genes are instructions, and proteins are functional structures that can do stuff. So we've got very few genes, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, something like that, genes, which is way small. Humans have 30,000 plus genes. So the genome is way simple compared to our genome, and our genome is actually quite simple compared to plant genomes, believe it or not. Anywho, here's that RNA wrapped up inside, but kind of the most conspicuous part of the virus are these spike proteins on the outside. And the function of those spike proteins is to fit like a key into a lock onto this ACE2 receptor of human cells. So proteins have a size and a shape and electrical properties which allow them to interact with molecules that have complementary size and shape and electrical properties. So I'm going to oversimplify it here, but let's say this knob right here has a positive charge right here, and this socket right here has a negative charge right here, and they fit. If they rub up against each other, this is going to lock into this. When that happens, it's going to send a signal to this human cell to sort of engulf this whole particle right here, which is the virus. So here's how that happens. Excuse me. Here's the virus particle right here. The virus particle comes and these spikes lock to these receptor proteins, these ACE2 receptors, and then a process called receptor-mediated endocytosis happens. Receptor is these sort of receivers, these antennas on the outside of the cell. To mediate means to help. Endo means inside, and cytosis means of the cell. So receptor-mediated endocytosis means engulfing something from the outside of the cell to the inside of the cell with the help of a receptor. So that's how the virus gets inside of your cell. And these receptors, these ACE2 receptors, are found in your spleen, they're found in your heart, they're found in your intestines, and most importantly in this case, they're found inside of your lung cells. Okay, so once that genetic information gets in there, what goes down? Well, normally inside of human cells, this is what happens. Human DNA, through a process called transcription, is made into a copy of human RNA. Yes, humans have RNA also. And then that RNA, through a process called translation, you might remember, is used to assemble a string of amino acids. And amino acids are like these relatively small molecules. There's 20 different kinds, and they all have a different size, they all have a different shape, and they all have different electrical properties. So depending on what order you link them together in, so these are amino acids linked together in what's called a peptide, they're going to interact with each other. Like, for example, if this one is attracted to this one, which is to say this one is attracted to this one, these two right here, that's going to cause a bend. So due to attractions and repulsions, this chain of amino acids folds into a shape which allows it to do a certain function. For example, these skizzers right here are of a certain size, a certain shape, and a certain material that allows them to do a job, which is make telephone calls to your friends. Coronavirus, on the other hand, hacks your cells to do this instead. Viral RNA gets into your cell through receptor-mediated endocytosis, which we looked at a moment ago. Once that viral RNA gets into your cell, two different things can happen. The first is translation. Ribosomes, which do translation, are built to read RNA. And I have this labeled viral RNA, but in real life, RNA is RNA is RNA, just like DNA is DNA is DNA. When I say viral RNA, I'm saying it came from a virus, but your cell can't tell where it came from. All your cell can tell is that it's RNA, and it's going to follow the instructions on the RNA. When it does that, it's going to make viral proteins, not human proteins. Viral RNA is used as instructions to create viral proteins by the ribosomes in your cells. That's the first thing that's going to happen. The second thing that's going to happen is this. That viral RNA is going to get replicated. So our cells do not have enzymes to take RNA and make an RNA copy of it. Our cells do have enzymes to make a DNA copy of DNA. That's called DNA polymerase. But we don't have enzymes to make an RNA copy of RNA. So those enzymes actually are right here. So this has instructions for how to make a special enzyme that humans don't have, and that enzyme can replicate viral RNA. So here it is making 1, 2, 3, 4, 5 copies of that viral RNA. In real life, it would, I don't know how many it would make, but probably a buttload. So anyway, the viral proteins, including that enzyme that does this replication, but also those spike proteins and the membrane proteins and all those proteins that we looked at for those 13 genes we looked at earlier that make the 13 proteins. That's what we're talking about right here. And here's a bunch of copies of that viral RNA. So the virus uses our endoplasmic reticulum and Golgi apparatus to assemble viral proteins with viral RNA. And what do you get when you assemble viral proteins with a viral RNA? That's right, you get new virus particles. So the viral protein and the viral RNA go to make new virus particles. Your cells become virus particle manufacturing machines. So here's that same process in a different model. Here we are with coronavirus, SARS coronavirus 2. And here it is attaching to that ACE2 receptor on a human cell right here. Once it gets in there, the RNA gets released. The enzymes that are found inside of the virus make a bunch of copies of that RNA. Our enzymes, our ribosomes, take the RNA and use the instructions in it to make viral proteins. And then those two things are combined in the Golgi apparatus to make new viral particles, which go to erupt from the cell to go infect other cells in your body and in other people's bodies. So here's that process. Virus gets into a cell, your cell. Your cell makes a whole bunch of copies of that virus. And the act of doing that can and does kill your cell, thereby releasing a whole bunch of particles to go infect your other cells, and when you cough, to go infect other people. So that's not the only way that the virus kills your cells. Next time, we're going to look at the pathophysiology of this process. So we're going to look at how exactly it kills your cells and how exactly it causes disease. And I know that might sound boring, but it's actually going to be super sweet. So stay tuned. I really miss you guys, and I'll see you next time.